Have you ever wondered how nature performs incredible feats of chemistry with such precision and speed? The secret often lies in enzymes, nature's tiny biological catalysts. But what if we could create artificial catalysts that mimic these natural wonders, yet offer even more advantages? Get ready to dive into the fascinating world of nanozymes, where cutting-edge nanotechnology meets biology to unlock revolutionary possibilities. Imagine materials so small they're measured in nanometers. That's 1/1000th of a meter. Nanozymes are precisely these. Nanomaterials, typically ranging from 1 to 100 nanometers in size, that possess remarkable enzyme-like properties. They can accelerate specific chemical reactions, just like natural enzymes do, but they are not proteins. This unique class of materials beautifully combines the best features of traditional chemical catalysts and biological enzymes. They offer the robustness and tunability of inorganic materials with the high efficiency and specificity often associated with biological systems. This makes them incredibly versatile tools for a wide array of applications. The concept of artificial enzymes has been explored for decades, but the specific term nanozyme and the recognition of inherent enzyme-like activity in nanomaterials are relatively recent breakthroughs. It's a story of scientific curiosity leading to groundbreaking discoveries. The term nanozyme itself first emerged in scientific literature around 2004. This was used to describe the catalytic activity observed in certain gold nanoparticles. Researchers noticed that these tiny gold structures could facilitate reactions in a manner reminiscent of enzymes. A pivotal moment arrived in 2007 with the discovery concerning iron oxide, Fe3O4, nanoparticles. Scientists found that these common magnetic nanoparticles exhibited intrinsic peroxidase-like activity. This meant they could catalyze reactions similar to the enzyme peroxidase, which is crucial for many biological processes. This discovery truly ignited the field, demonstrating that the catalytic properties were inherent to the nanomaterial itself, not just a surface modification. The world of nanozymes is diverse, encompassing different strategies to achieve enzyme-like functions. Understanding these categories helps us appreciate their design principles and potential applications. One type involves nanomaterial hybrid enzymes. In this approach, natural enzymes or specific catalytic groups are carefully modified and integrated onto the surface of nanomaterials. The nanomaterial acts as a stable scaffold, enhancing the enzyme's stability, activity, or even its recyclability. This hybridization often protects the delicate enzyme from harsh environmental conditions, allowing it to function effectively in settings where it might otherwise degrade. The second, and perhaps more revolutionary, type consists of nanomaterials with inherent enzymatic catalytic properties. These are the true nanozymes in the purest sense. Here, the nanomaterial itself, due to its unique atomic structure, surface chemistry, and nanoscale dimensions, intrinsically displays enzyme-like activity. They don't need an attached enzyme. They are the catalyst. Their mechanisms often mimic those of natural enzymes, involving active sites, substrate binding, and the lowering of activation energy for specific reactions. This inherent activity is what makes them so exciting and distinct. While natural enzymes are incredibly efficient, they come with certain limitations. This is where nanozymes truly shine, offering compelling advantages that make them highly attractive for various applications. One significant benefit is their lower cost of production. Natural enzymes are typically proteins, which require complex and often expensive biological synthesis processes, purification, and careful handling. Nanozymes, being inorganic materials, can often be synthesized using simpler, more scalable, and less costly chemical methods. This economic advantage makes them more accessible for large-scale industrial applications. Another critical advantage is their greater stability and durability. Natural enzymes are delicate biomolecules. They are highly sensitive to changes in temperature, pH, and the presence of organic solvents, or proteases, enzymes that break down proteins. Nanozymes, on the other hand, are generally much more robust. They can withstand a wider range of temperatures, extreme pH conditions, and harsh chemical environments without losing their catalytic activity. This enhanced stability allows them to be used in conditions where natural enzymes would quickly denature and become inactive. Furthermore, nanozymes offer tunable catalytic activities. Unlike natural enzymes, whose activities are largely fixed by their genetic code, the catalytic properties of nanozymes can be precisely engineered and modified. By altering their size, shape, composition, surface modifications, or even their aggregation state, scientists can fine-tune their activity, specificity, and even their multi-enzymatic functions. This tunability provides an unprecedented level of control over their performance, allowing them to be optimized for specific applications. Finally, nanozymes are often easier to store and transport due to their stability. They typically do not require refrigeration or specialized handling, reducing logistical complexities and costs. Their reusability is also a major plus. Many nanozymes can be easily recovered from a reaction mixture and reused multiple times without significant loss of activity, further enhancing their cost-effectiveness and sustainability. One of the most intriguing aspects of nanozymes is their ability to exhibit multifunctionality. This means a single type of nanozyme can mimic not just one, but often multiple enzyme activities simultaneously or under different conditions. This capability is rarely seen in natural enzymes, which are typically highly specific to one or a few reactions. A prime example of this multifunctionality is observed in iron oxide, FeO4, nanoparticles. These remarkable nanozymes can display both peroxidase-like activity and catalyst-like activity. Peroxidase activity involves the breakdown of hydrogen peroxide into water and oxygen, often in the presence of a substrate. Catalyst activity, on the other hand, directly decomposes hydrogen peroxide into water and oxygen without requiring an additional substrate. What's even more fascinating is that the dominant activity of FeO4 nanoparticles can be influenced by the surrounding environment, particularly the pH level. At certain pH values, they might primarily act as peroxidases, while at others, their catalyst-like activity becomes more prominent. This pH-dependent switching of activity highlights the incredible versatility and adaptability of nanozymes. This multifunctionality is incredibly valuable for complex biological systems or industrial processes where multiple enzymatic steps are required, potentially simplifying reaction pathways and improving efficiency. The unique properties and advantages of nanozymes have opened up a vast array of potential applications across numerous fields. Their versatility makes them powerful tools for addressing some of the most pressing challenges in science and technology. In the industrial sector, nanozymes are being explored for various processes. They can be used in bioremediation to break down pollutants in water and soil, offering an eco-friendly alternative to traditional chemical methods. In the food industry, they might be employed for quality control, detecting contaminants, or extending shelf life. Their stability and reusability also make them attractive for large-scale chemical synthesis, potentially replacing more expensive or less stable catalysts. The medical and biological fields are perhaps where nanozymes hold the most transformative potential. Their ability to interact with biological systems at the nanoscale opens doors to novel diagnostic and therapeutic strategies. For disease diagnosis and treatment, nanozymes are being developed as highly sensitive biosensors. They can detect specific biomarkers associated with diseases, even at very low concentrations, enabling early and accurate diagnosis. For instance, their peroxidase-like activity can be harnessed in colorimetric assays, where the presence of a target molecule leads to a visible color change, indicating disease. In vitro detection and in vivo monitoring, nanozymes offer unprecedented capabilities. They can be integrated into diagnostic kits for rapid point-of-care testing, allowing for quick and reliable results outside of a traditional laboratory setting. Inside the body, they can be engineered to target specific cells or tissues, providing real-time monitoring of biological processes or drug delivery. Their small size allows them to navigate biological barriers, reaching areas that larger molecules might not. One of the most exciting therapeutic applications is in tumor catalytic therapy. Many nanozymes can generate reactive oxygen species, ROS, under specific conditions, which are highly toxic to cancer cells. By designing nanozymes that selectively accumulate in tumor tissues, they can be activated to produce ROS, effectively destroying cancer cells while minimizing harm to healthy cells. This approach offers a promising new avenue for cancer treatment, potentially reducing the side effects associated with conventional therapies. Nanozymes are also proving effective in combating bacteria and biofilms. Biofilms are communities of bacteria encased in a protective matrix, making them highly resistant to antibiotics. Nanozymes can be designed to disrupt these biofilms or generate antimicrobial agents that effectively kill bacteria, offering a new strategy to combat antibiotic-resistant infections. Their ability to generate ROS can directly damage bacterial cell walls and DNA, leading to their eradication. Furthermore, research is exploring their use in leukemia therapy. Specific nanozymes are being investigated for their ability to induce apoptosis, programmed cell death, in leukemia cells, offering a targeted therapeutic approach. Their tunable properties allow for the development of highly specific agents that can differentiate between healthy and cancerous blood cells, leading to more effective and less toxic treatments. Beyond these, nanozymes are being explored for applications in environmental sensing, agricultural improvements, and even in the development of new energy technologies. The field is rapidly expanding, with new discoveries and applications emerging constantly. The journey of nanozymes, from their initial discovery to their current diverse applications, is a testament to human ingenuity and scientific collaboration. As students, you are at the forefront of a new era of scientific discovery. The principles of chemistry, biology, and material science converge in this exciting field, offering endless opportunities for innovation. The challenges ahead include further understanding their precise mechanisms, ensuring their biocompatibility and safety for in vivo applications, and scaling up their production for widespread use. However, the potential benefits are immense, promising breakthroughs in medicine, environmental protection, and industrial processes. Are you inspired by the power of tiny catalysts? The world of nanozymes is waiting for your curiosity and creativity. Dive deeper into this fascinating field. Research recent scientific papers, explore university programs in nanotechnology or materials science, and consider projects that involve these incredible materials. Share your excitement, talk about nanozymes with your friends, teachers, and family to spread awareness about their potential. Imagine the possibilities. How could you use nanozymes to solve a problem in your community or the world? Your innovative ideas could be the next big breakthrough. If you're enjoying this video, don't forget to hit subscribe and turn on the notification bell. so you never miss an update.